Well, good afternoon, or rather good evening, ladies and gentlemen. Also, I would like to thank the organizer for the invitation. And, oh, sorry, yes. and for the opportunity to have this presentation today. It's really a great pleasure for me to be here. Yes, as a start, I would like to provide you with some bits of information about Lonza. Lonza is a healthcare company based in Basel, and the annual sales in 2009 amounts to around about 2.7 billion Swiss francs. Lonza operates globally with currently 26 production and R&D sites and more than 8,300 employees. Lonza now is a global leader in custom manufacturing of active pharmaceutical ingredients, or APIs, of both chemical and biological products. And beside that, Lonza is a global leader in manufacturing of products for cell therapy. When I received the invitation to give this presentation today, at least for me, the question came up, where is the common ground of nanomedicine and Lonza? And so I took the time to read these roadmaps in nanomedicine published by the European Commission and the European Technology Platform on Nanomedicine. This roadmap was already presented today. And yes, I found that per definition, cell therapy is part of the area regenerative medicine with a strong emphasis on manufacturing of what's called product class 2, that means cells, and the related enabling technologies. And for me, that's a link to Lonza, and so I decided to talk about manufacturing of cells for cell therapy, and I hope this will meet your expectations. Yes, thanks to more than 40 years of experience in normal human cells and cGMP compliance, Lonza became the industry leader in development, manufacturing and commercialization services for cell-based therapeutics. And today we partner with our customers to develop client-specific customer protocols supported by the expertise of our process development department. The main production sites for cell therapy products of Lonza are located in Walkersville in the United States and in Singapore. As already mentioned, Lonza is globally leading in custom manufacturing, and therefore we are extremely experienced in bioprocessing technology. And our vision is now to bring this bioprocessing technology to cell therapy to meet the commercial needs of our customers, or in short, to make cell therapy happen. Why is this our vision? Why are we convinced that this is a good way to go? What we learned during the last years is that there's a growing need for cells, both at therapeutic and research grade, that can be manufactured in scalable processes. And we are convinced that this need for manufactured cells in large quantities and reproducible quality will rise during the next 20 years, since the corresponding markets will evolve. And those markets are therapeutic markets, research markets, as well as cell-based pharmaceutical screenings markets. And well, yes, our experience is supported by hard facts. We discussed already the aging population of Europe, so we can skip these figures. Against this background and based on further facts and aspects, market projections for cell therapy products predict sharp increase. For example, this analysis, stem cells, hypes and hopes, prospects and strong increase of the market for stem cell therapy products from nearly zero in 2005 up to over $8 billion market in 2016. So what can we do to address this growing market? In 1970, the first paper was published on cultivating mesenchymal stem cells isolated from bone marrow. At that time, those cells were called fibroblast colonies. However, this was the first time that it was published that adherent growing cells can be isolated from bone marrow. Well, obviously, we have come a long way since then, and with major advances in quality and quantity of the product, but there's no significant breakthrough since then in manufacturing of adherent growing cells. So during the last 40 years, the processes were enlarged, refined, improved, and now the purity quality of the product corresponds to GMP levels of pharmaceutical products. Just to give you a picture, here you can see the 10-layer stacks where the cells are cultivated and how the medium is removed from these containers and replaced by fresh medium from these sterile bags. So Lonza manufactured those cells at unprecedented scale, but this won't be enough in the future. So what about upscaling? We could elongate this queue of employees, but I think you agree that wouldn't be the smartest solution. So what's next? So it's important to know that the production processes determine the properties of the cells, and so the options are limited. We could show at Lonza that an enlargement from 10 layers stacks to 40 layer stacks is possible. For example, cells grow to same density. But those containers are difficult to handle, and this solution is not elegant but manageable. But also, even this 40 layer stacks won't be sufficient in the future. So, and then that's the question. And I think this is a point where cell therapy meets nanotechnology. So, according to the needs I presented during the last minutes, I think that one approach could be the development of intelligent surfaces, for example, for the bioreactors, that could help to manage, for example, attachment or detachment of cells on demand, for example, or to control the growth conditions of the cells. That means proliferation or differentiation of the cells, or that could transmit signals into the cells, for example, to simulate a dense cell layer to single cells. And I'm sure that there are many, many more options. This is just a short list of potential ideas how the production processes could be improved and especially enlarged in the future. So the opportunities of those future nanotechnology solutions are, as I already mentioned, the control of cell expansion rates, differentiation, and the phenotypes. And those materials could also help to, for example, to deliver drugs or biosensors into the cells, or to allow for cell delivery designed to selectively degrade via command, that means beyond the cell manufacturing processes. And risk and hurdles, as always, the timelines. We all know that it needs time to develop those materials from the R&D stage to marketable products. Safety issues were already discussed today. And also cost is an important factor, since those intelligent materials have to replace good enough materials. So finally, I can say we need your input. We need the help of nanotechnology to develop new systems that will allow for scalable manufacturing of cells for cell therapy in the future. Thank you very much.